Yeah, first note, of course, the meeting is being recorded as always. So if you haven't made it so far, you can also watch the recording later. But in any case, I'm very happy to introduce Riccardo Fusi today. He did his bachelor's and master's in agricultural sciences in Italy at the University of Milan and Bologna. In Bologna, he later on worked also with Silvio Salvi, in which we had here in this meeting, I think maybe at the beginning of this year or last year. So some might be familiar with him. Then Rick did his PhD in Nottingham with Malcolm Bennett and Jonathan Litch, where he now also works as a postdoc. And he works on genes controlling deeper rooting using genomic and phenomic approaches. And I think that's exactly what he will talk to us about today. The stage is all yours.
Thank you so much, Gwen, for the nice introduction. As Gwen mentioned, since my master, I've been working on identifying and characterizing genes regulating root development. And I will talk a little bit about that today. And first and foremost, thanks to all the IBH crew for inviting me for this seminar and hosting me and kind of like hosting me and messing around with all my computer failures. But it's a very honor for me to be here and speak to all of you. And this work has been mainly funded by BBSSC and ARPA-E, US environmental agency company. And I really want to say thanks for them to funding all of this work. Next one. So as we all are experiencing nowadays, climate changes, challenges really are affecting us. And I just put here a snapshot of this year, but is really what's happening over the past decade, probably. The severity of drought and floodings events are really start to kick in. And in a very renowned rainy country like the UK, as we have here, here we have experienced one of the droughtiest spring in the past 70 years. And most of the reservoirs of our basins have fallen beyond the critical level where the government has to take critical action about water usages. So this is really touching us not only by this, next one, but also what's happening in the fields. Oh, wait, go up one, sorry. So that's also what's happening in the fields. As you can see, these are some field trials that we had here in Nottingham from last year, where like the very low moisture content really affected plant establishment, and thus really is going to really impact our food security in the coming year. So really tangible effects of this. Next one. So basically, I think it's, of course, I'm speaking at the Barley Hub, so I think it's good to remind everybody that barley is very important crop and is one of the most ancient one of the first that got domesticated in the Fertile Crescent more than 10,000 years ago alongside wheat. And one of the most tolerant crop that is able to grow in the most difficult environments. And that basically creates a very nice variability amongst all of the population that we have available for study the genetic variation of these adaptive traits. Is the first most relevant crop for production as a simple diploid genome, and is now, I think, is quite widely considered a model crop for genetic studies because of this, compared to a little bit more complex wheat, and is also easy to transform. Next animation. But however, very few genes controlling root resilience has been identified and characterized, and that's what I will talk about a little bit today. So roots are quite complicated to study because they grow in soil. Soil is really an opaque matrix and is generally hard to study. You have to go in the field, do a lot of shovel a lot of shovelomics is really hard work. And this, all of this hard work in the past few years has been basically put in place to discover root architectural and anatomical traits that can be of advantage for roots in exploring deeper soil layer. It's in fact important exploring deeper soil layers because water and nitrogen, which are two of the most important macronutrients that the plant uses for growth, they are present in the very deep soil layer. So having a steeper root angle is very, very important to increase plant resilience to water stress. Next animation. But it's not only that is important, it's also important to have a cheap root system. So you cannot invest too much biomass in growing deeper. You also need a cheaper system to do so. And alongside those traits, basically high root cortical parenchyma, cortical cell size, and low cortical cell size have been identified in regulating carbon deposition and optimizing carbon deposition to allow the plants to still perform in terms of yield. But another trait that I will talk about today a little bit is also how xylem vessels, more and narrower xylem vessels are important for this. And I will talk a little bit about xylem vessels, that they are very important for water uptake. Next one. So what I will talk today a little bit is about how we exploit Tilmor. Tilmor is a tilling population that has been developed in Bologna by Silvio Salvi group, and how this helped in identify two key genes regulating root architecture and root anatomy, especially two genes regarding root architecture EGT1 and regarding root anatomy PIN1. Next one. So just giving a little bit of introduction of what Tilmor is composed of. So Tilmor is a very broad population of sodium azide-based mutagenized population in Morex. It contains more than 3,500 M6 lines with a very high mutation frequencies. And using forward genetics screenings has been able to provide more than 60 root mutants. And this currently has been rescreened, as I'm aware, and is probably more. This number is likely to be higher than this. Next one. So basically, what we have is that we have a list of different phenotypes that have been identified: shoot and roots phenotype. Next animation. However, we do have what I will talk about today. We'll put some focus and emphasis on one mutant. So this basically, overall, this population have provided two key genes regulating root angle in cereals. One is EGT2, which has been discovered by Gwen and the Silvio Salvis group, and has been characterized by Gwen, as well as EGT1, which alongside Gwen and the Silvio Salvis team, we characterized this gene EGT1 regulating hypergravitropic roots in cereals. And I will talk a little bit about a little story about short root mutants that we have been discovered, and we also discover to carry important regulation into the vasculature development. Next one. So, as I say, root angle, it is very important is a very important trait to study. However, the genetic of it has been quite vague, especially in cereals, and not many genes are known to regulate this trait. It allows the uptake of water and nutrient from deeper soil layers, as well as enhancing carbon deposition in deeper soil layers. However, investigating this in real soil, you can use different methods. And here I propose an undisrupted method that we have been using in the past using X-ray CT that help us in characterize it. Next one. So I think everyone knows X-ray, the X-ray CT facility that we have here in Nottingham, state-of-the-art facility that we are using for soil and roots characterization. IB used this one extensively for root angle. This technology allows non-invasively visualization of an interior of a solid object in a 3-dimension, and basically based on different density of different objects or different feature containing an object, we are able to segment out roots and its trait, including root angle. Next one. So this one helped as I said, we set up experiments here in Nottingham on 10 days old seedlings, as you can see in the panel A, and one of the mutants, TN194, which is basically a mutant in EGT1, it has really steeper root angle. Is also bend faster, which is again suggesting about regulating somehow to be involved in regulating the pathways of the gravitropic machineries that we'll talk about a little bit about later. And this happens not only in the seminar roots, but also in the crown roots, as you can see from panel B. In mature plants, crown roots again show steeper root angle, and lateral roots as well show the same phenotype, suggesting that EGT1 regulates root angles in all the classes. Next one. So with Silvio Salvi in Bologna, we have been doing bulk segregant analysis. So what we have been doing was basically taking the segregant population resulting from the F2, and we had 2 pools, gravitropic and normal shallow roots. We harvested DNA samples and we sent it over for exome capture. And basically what we found is that 2 alleles, TN194 and 3580, they carry mutations in the same gene, which is in chromosome 6 of barley. One of them is a splice receptor variant 3580, and the other one is TN194, which is a missense mutant, which also caused by protein modeling disruption in the function of this domain, suggesting that might be causative of the phenotype that we observe. But we expanded this research also by looking at land races, and what we see is that we see 7 haplotypes to be identified within this pool, and especially two of the haplotypes, haplotype 2 and 4, they exhibit differences in root angle. A narrow root angle is showed in haplotype 2, and a shallower angle is shown in haplotype 4, as also you can see from the graph below. So this suggests that there might be interesting for breeders to breed for this gene, because variability it exists. And is also like we also observed differences in protein structure in haplotype 2. So next one. So we started to have a look into the how this gene regulates this response to gravity, because the bend super fast, so something must happen. We firstly look into downstream genes that they are differentially regulated, and the 2 most important clusters were related to ROS processes and cell wall biosynthesis processes. We then decided, I then flew to Matthew Tucker in Adelaide to run in situ hybridization as leading expert there, and basically what we saw is that despite you can see that it's kind of expressed like in all the root teeth from a longitudinal section, it is a little bit higher in the elongation zone. And when we did radial cross sectioning throughout all the all the root, we see that especially in the elongation zone, it was highly expressed in the steal cells, suggesting that something might going on in regulating this in this tissue type. So next one. So what we did next was see like what were the effect in the ROS production, and what we saw is that especially in the early stages in the root tip, the ROS were lower in EGT1 compared to Morex, suggesting ROS production to be affected. And then in order to confirm, next one, in order to confirm how this impacts the cell wall organization and biosynthesis, we decided to test the stiffnesses of different tissues and in different developmental stages, and we especially targeted the elongation zone. In this case, early, middle, and mature. A bit more mature elongation zone. We divided in 9 zones. And what we observe, interestingly, is that the effect on the cell wall, on the cell wall properties such as stiffness, they were more pronounced in the cortex compared to the steal, suggesting that there is some sort of movement or crosstalk of these genes between the two different tissues in the root. So next animation. What we saw, what we proposed, I will not too much detail, but basically what you have, one of the main basically driver or root growth in soil is regulated by two machinery. One is the gravitropic machineries that regulates the gravitropic response, so the root growing downwards, and the anti-gravitropic response that basically regulates the root to grow in a more horizontal way. We propose EGT to regulate this latter process. And this is via crosstalking between different tissues, by regulating the peroxidases and the cell wall loosening enzymes, and different stiffening the cell wall in these two compartments. So basically, by softening the cell walls, then the root is freely able to just grow downwards. And this is a subject of Raoul Bosley grant as a new investigator grant. So this work has been is currently this research in understanding this mechanism is currently taken over by Raoul in his grants, which I think he spoke here quite early this year. Next one. So as I mentioned, the second story I will talk about today is a story that led to the characterization of the short roots. Phenotypes that we observe in Tilmore, and how this regulates vasculature patterning. And this is what I will talk about in the next slides. So similar to what we did in EGT1, we started from a pool of roots that were shorts, collected the DNA, and we run exome capture on it. And what we observe is that we had basically the formation of 3 alleles. Interestingly, we have 2 in the Tilmore population. We have TM5141 and 5992, which they are missense and again missense and splice variant in different exons and in different position. And we have another allele which was present in one of the IPK tilling population at the time in Barke background. This is marked as TB is the tilling Barke, and TM is the Tilmore mutants. So next one. So this was what we identify was a gene in this case that is member of the PIN family, which is very well described in Arabidopsis in regulating polar auxin flux. Auxin is the most important hormone in regulating growth development in all plant organs, especially also in roots. And basically what PIN1 does is regulating the downward flux through the xylem of auxin to the QC. And in Arabidopsis, we only have 1 family member of the PIN1. However, in Sirius, we have 3 members, PIN1A, PIN1B, and sister of PIN1, suggesting some level of subfunctionalization of these genes. Sorry, I have some. Next one. So we started to have a look at the root tips, and basically what we did simply was just using propidium iodide and see how they look under the microscopes. And we saw that as simple as it was, the root apical meristem was shorter and with more proliferation of root rays, kind of like indicating like different and imbalances in root homeostasis in the root tips. What we wanted to understand was like how the dynamics happen and how different dynamics were present on a cellular level. So what we did, we optimized clear C protocol developed by Nico Gellner group in Lausanne. And basically, we were able to visualize, have a cell-by-cell resolution under confocal microscope. And what we saw is that this mutation affected cell division in the root tip. And next one. This was not only in one allele, this was also in all the other allele like exhibit similar phenotype. So all root tissues were affected by this. However, what we did was starting to observe like some weird ectopic differentiation in the middle central metaxylem, which we were not really understanding what was going on by then. So next one. So what we decided to do is running a like cross-section instead of having a longitudinal view of it. We decided to have a radial view of it to understand better what was going on. And basically, if I don't know if I think you well know how a barley root section looks like, but I will just run quickly through it. And basically, what you have in seminar roots is a central metaxylem that runs in the middle, and you have all the peripheral metaxylem that surrounds the central metaxylem, and they are all kind of like randomly, not randomly, sorry, they are kind of very well organized radially around the central metaxylem. And they are enclosed by the endodermis, which basically enclose all the steel part, and then you have the cortex on the outside. This is on Morex, on agar, and basically what you can see here is in blue, when I stain it with, I have a double staining here, in blue with cacofluor white, and in yellow with basic fuchsin. So what you can see here is the yellow markers of lignin and suberin deposition. So you can clearly mark the central, so the xylematic level or the highly lignified organs. And next one. And this is what we see in the Morex, and this is what we see in the mutants. So in the mutants, when we cross-section it, we observe this very randomly disorganized ectopic differentiation of like peripheral metaxylem, as well as we see this very highly lignified, what we decided to call inner parenchymatic steel layer. So it's the first layer surrounding the central metaxylem, and is highly lignified. It is highly lignified, and also the cells are severely enlarged. This was not the case. This was the case for the mutants in Tillman population, as well as in the Barker population. Next one. So what we basically like all the experiments were done in agar at the time, but I was also interested in looking at the soil and see whether these phenotypes are also conserved in the soil matrix. And likely so it was the case. So we use X-ray CT to confirm the short root phenotype at 9 days old, as well as the anatomical phenotype. However, the soil caused restriction in the root diameter, so this was not exactly entirely visible because the effect of the soil matrix. The vasculature patterning defects were still maintained in the mutants. Next one. And because of course the nature of the gene is a PIN1 being a auxin efflux carrier, we decide to run a classic pharmacological assay by imposing stress with NPA. So NPA is basically a auxin analog that goes into the efflux carrier, and it just perturbs its functionality, not allowing the auxin to go, disrupting basically the polarity of the distribution. And when I apply increasing concentration of NPA, I went a little bit mad here, but you can start to see the effect after 5 micromolar of NPA into the vasculature tissues, suggesting that you really need a PIN1 gene to control vasculature patterning. Next one. And on the other end, we decided to add auxin to see whether there was an eventual possibility that the lack of auxin at this cell layer was causing the phenotype. So we impose auxin in the mutants as well as in the wild type. However, we didn't see like any differences between the mutants under NAA and the MOREX, suggesting that you still need a functioning, fully functioning PIN1 gene to regulate this process, not auxin alone that regulate this. Next one. So interestingly, now I'm gonna shift a little bit. I hope you can forgive me. But we also have the possibility of looking into brachypodium mutants. So we were also interested in see whether this was conserved among different cereal species. So brachypodium is another model crop, a little bit simpler than barley, and a bit more compact. And is kind of widely used also in plant among plant scientists to characterize gene functions. And at the time, Devin O'Connor has these mutants. We collaborate with him at the time, and we see that despite there was not a severe short phenotype, probably due also to the high mutation load or different functions of these genes in brachypodium, we still see defects in the central still in the inner parenchymatic still layer cells, that they were enlarged and highly lignified, suggesting that this process is also confirmed across species. And on top of it, we also have access to not only the PIN1 member, but also other PIN1 family member. And we tested it in PIN1B1 mutants. What we see is that this is not showed in the PIN1B. That suggests that PIN1A specifically is a key regulator of this process among its family members. Next one. Because as I mentioned before, we do have access to brachypodium resources. In brachypodium, we were also developed reporter lines in different genes in the different members of the PIN1 family, and you can see like a classic DR auxin response expression of DR5 in red in the left, and then you see the PIN1A reporter line, PIN1E reporter line, and sister of PIN1 reporter line. And you can see the expression in the vasculature patterning where PIN1B kind of overlaps. However, sister of PIN1 has a very specific localization. Likely, in this case, we didn't have market, so it's likely to be in the protoxylem or in the peripheral metaxylem, but very specific expression, then suggesting that there is a lot of crosstalk between these genes. Next one. So, I'm basically what I did at that point was like I wanted to understand what were the physiological impact of having such a perturbed vasculature patterning. Is it beneficial in draw stress? Is it is it bad in draw stress? So I started to play around a little bit with draw stress experiment, imposing draw stress experiments in brachypodium plants, and try to understand whether we see differences at physiological level. I do have an incompleted work at this time that I will not present here. But one of the nice results that I have from here is that we have been starting to screen also the shoot at the time because we were very much focused on the root, doing everything on the root here in Nottingham. But we decided to have it a go and see also if these vasculature defects were also conserved in the apical part. So because I was running those experiments, I decided to chop few samples out and handle it to our lab experts here in Nottingham who helped me in imaging those stems. And next animation. And as you can see, in this case, I'm just going to show the in well water situation. We can clearly see that these defects were also present in the steel part. I cannot really say anything about the leaves because they are very tiny and I couldn't do any characterization. But for the steel level, we still see some differences in vasculature patterning even at this level. And next one. And of course, I had a look. In this case, I've been using mature roots that we never had a look at really. And at least in brachypodium, we couldn't really quantify it, but just in a descriptive way, we observed again that the organization was highly disrupted. However, we didn't see really much differences in terms of size or we couldn't quantify it at that stage. So yeah, this is basically it. So I just close with like few key points of the talk. So the first one is I hope that I probably alongside many other collaborators of this project, I kind of like highlighted how Tilmor revealed to be a phenomenal genetic resources to map and characterize genes, regulating fundamental serial root process, and then how EGT1 controls root growth angle via regulating the anti-gravitropic offset machinery, as well as PIN1A uniquely regulate vasculature development in cereals. However, I think one of the biggest point is like testing how these genes will really affect plant on a physiological level would be the next stage of our research at this time. So I would like to conclude. So just to quickly acknowledge everyone, I really want to say thanks to the group in Nottingham here, especially Malcolm and Raoul, which basically guided me through all of this research, and Sasha Mooney, Craig Starrock, and Brian, which they are leading expert in X-ray, CT, and soil physics, as well as from Bologna, all the Silvio Sarvis group that helped me with the gene cloning and identifying those genes, as well as mention to Jonathan, which is now hopefully enjoying his retirement, but has been a vital mentor as well during my PhD, as well as Matthew to have hosted me in Adelaide for the in situ of EGT1, and all the Campus Biomedico in Roma that helped me with the raw staining, and all the people in the IPK that helped me with all the resources in the bark population for the PIN part, as well as all the bulk segregant analysis that they have done there. And yeah, that's basically it.
Cool. Thank you very much.
Yeah. Sorry everyone for the skipping and doing.
That was surprisingly more stressful than expected. So we have some questions in the chat. Let's start with that. So Laura Rossini is asking, is first saying, thanks for sharing your very nice work. And she says in rice, she thinks that PIN1 mutations affect also tillering. And did your mutants have any tillering phenotype as well?
So that's something. So that's something that we observed. We observed in Brachypodium. In barley, I never got the plants that old because of the nature of the experiment at the time. But in brachy, I was able to push them a little bit, and I saw some effects on tillering in roots. But again, like I saw something in Brachypodium. Brachypodium, I think, is closer to rice than barley, so it would make sense.
Yeah, so if you have more questions, please put them in the chat. Otherwise, I will now go ahead with my questions until I find more. I'm, of course, curious in EGT1 because we also look up for the EGT2 if there's any natural variation, and we didn't find any. So for your EGT1, the alleles in the natural variations, like the natural variations in the different alleles, what did they do on protein level? Was it
so? I need to have a precise look at what, because the modeling was done by collaborators in the vet school. But as I remember, there were some changes in the polarity of the protein. I don't remember which protein specifically was it, but it was a change, amino acidic change that changed the polarity somehow, which basically caused like the pocket within the tabby domain, if I'm correct, to be affected. So likely to change transcriptionally later on. Okay. But I can retrieve that information more in detail for you. But we have done that analysis. We have it. I can retrieve it.
And then a question more related to the mutant population. So you chose short root mutants for your analysis, but I assume that a lot of mutations in important genes make short roots, right? Like once you disturb anything, the phenotype is probably to make it shorter. Was there anything special about the ones that you choose?
No. They were just simply not that I think so. I think it was something also related to rootase at the time. I think it was a combination of short root and higher rootase at that stage. But yeah, not that I was nothing like at the time. We were not expecting. There was something in literature. So it was suggested by some PI to have a look, if possible, at the vasculature, because there we do have Professor Tony Bishop here. Is an expert in vasculature, and he characterized vasculature development and auxin distribution. And he mentioned that he observed changes in one of his manuscripts in the PIN1 mutant for the vasculatures. So it was a combination of things, I think, at the time that kind of led us to have a look at it.
Cool. Yeah, audience, if you have more questions, please put them in the chat. Otherwise, I have one more. You mentioned you had the Morex allele from the tilling population from your PIN, but you also had a Barke allele. Where did you isolate that from?
That was IPK. That was from collaboration from Niels and Sylvia.
So that was after you found that the mutation in the one is the causative one. Okay.
Yeah. Yeah, it was I think it was there were talks between the groups about bug segregant analysis, and probably the talk carried over, as always goes in our field. But yeah.
All right. I think we will conclude for now then. Thank you very much for audience for coming and listening. Thank you, Rick, for giving the seminar. And I was told I should announce that this is the last IBH seminar before the summer break, and we will go on Thursday, the 4th of September. I wish you all a great summer. Until then.
Thank you. Thank you, Gwen. Chat later.
Yeah, bye-bye.
Oh, thank you, Malcolm.
Yeah, thanks very much.
Thank
We'll
you, Malcolm, for it. Cheers.
No problem. Okay, we'll be in touch. Thanks a lot.